Information is the same, but we are going to go in greater depth of it in this lecture. So why is infection control necessary? Well, because we deal with things that we can't see that are dangerous to our health. Some of those things that we can't see include HIV and AIDS, tuberculosis, antibiotic and antimicrobial resistant organisms, and also hepatitis. These are the different types of diseases and pathologies and conditions that you will be faced with as a technologist. So you guys are familiar with the term medical asepsis. You should know it by now, right? Mm-hmm. As free as possible from any type of pathogens. What is a pathogen?
Something that causes disease.
Something, okay, so it's a microorganism that can cause disease, illness, or even death. So medical asepsis is trying to eliminate these pathogens from the environment as much as possible. Now, again, as free as possible. But are we completely eliminating those microorganisms? No, right? You look at a spray bottle, you look at the back of a dial, you look at the dial soap, you look at the Purell. What does it tell you? What's the percentage of elimination of germs? It's going to be 99.9%, so there's still that 0.1% that still exists. So, again, medical asepsis condition of reducing the probability of infectious organisms. The fewer organisms the healthcare worker is exposed to, the better the chance of resisting. But it also works the other way around.
Immunity.
Okay? Medical asepsis is to protect us, but we're also protecting who? The patients. Okay? Because we are the biggest enemy of the transmission of these microorganisms. So we eliminate some of these bugs through microbial dilution, the process of reducing the number of organisms accomplished at several levels. First one here is simple cleanliness, including proper cleaning, dusting, linen handling, hand washing techniques. By the way, how long should you wash your hands?
15 seconds.
Happy birthday. So some of you guys remember the lecture. So what is that in seconds? About 20 seconds. Okay, so you hum happy birthday twice in your head. Again, you don't do it out loud. People are going to think you're crazy. So you do it for 20 seconds. And the elimination of those pathogens is both chemical, soap, and then as well as mechanical, friction. Okay, so that's simple cleanliness. Second one here is disinfection. It involves the destruction of pathogens by using chemical materials on inanimate objects. And again, remember the difference between disinfectants and antiseptic. Disinfectants you use on inanimate objects, whereas an antiseptic you use on something biological. So mouthwash, that's not a disinfectant. That's an antiseptic. Okay? And then you're not using Listerine to wipe down your table. Okay? You're using 409. Okay? So disinfectant, inanimate objects. Then you have antiseptics for biological subjects. And the last one here, level 3, is surgical asepsis, but more specifically, sterilization. So what's the difference between sterilization and medical asepsis?
Sterilization completely removes all pathogens.
And what else?
And good bacteria as well.
So sterilization, you're on the right track. So sterilization is elimination of those pathogens, but you're also getting rid of their reproductive spores. Okay? So sterilization is elimination of those pathogens and their reproductive spores. Okay? And when we sterilize an object or some kind of instrument, it has to be wrapped, special type of wrapping to identify it as it being sterile. Okay, any questions? So how do you guard and prevent transmission infections? So we talked about medical asepsis. Then there's that term standard precautions. Standard precautions is also known as universal precautions, and you guys should know this by now also. So what does that mean? You treat everybody as if they have some sort of transmittable disease, bloodborne disease. Okay? So in other words, never ever let your guard down. Always have those standard precautions. They have some kind of disease. minimum requirements to protect yourself from your patients is what? What are the minimum requirements? What are you going to wear?
Barrier.
At least gloves, okay? Now, you will see, you will observe that what I tell you, what I share with you, is going to be completely different than what you see out there in the field. Okay? People get lazy. People get lackadaisic. Okay? You know how many times I've been to this restroom here and people are coming out not washing their hands? And this is a medical building. Right? People should learn how to wash their hands, but they don't.
They're preserving water.
They're preserved. Is that what you And one of the things I hate the most are those hand dryers. Okay? Because although you wash your hands and you dry it, that doesn't eliminate that handle being clean, because there's going to be somebody who didn't wash their hands grabbing that handle getting out of the bathroom.
They should switch that soap.
Huh?
No, Professor Brown said that the automatic dryers have actually more germs on them than the
They're nasty.
they're
Yeah.
gross
Yeah.
dryers.
All right, so standard precautions, that's universal precautions. Treat everybody as if they have a disease. Hand hygiene, you wash your hands for at least 20 seconds. Well, but what's the problem with constantly washing your hands?
Dry.
They get dry, so what are you gonna do?
Lotion.
Okay, so moisturize it, okay? Moisturize it. Keeping your work area clean, disposing of any type of contaminated waste material. This is how you prevent transmission of the infection. So basic types of acquired infections, the basic term is called nosocomial infection, which is an infection that you acquire at the hospital during some sort of procedure or during your stay there. So question is it possible that the patient is admitted for one type of illness and acquire another illness during their stay?
Yes.
Okay, from who? Who is it from? It's from us. Who else? It's also from the patients. Okay, so that's a nosocomial infection. More specifically, iatrogenic infection is acquired during treatment or a therapeutic procedure. And then community acquired just refers to the infection that you get outside in the community. Okay, so I get sick from Daniel. I get sick from going to Nordstrom's. I get sick from going with my kids to the playground. That's community acquired. Okay, so those who enter the facility may already have a community acquired. They go into the hospital for treatment, and then they can catch something else that's nosocomial now. Factors that promote nosocomial infections includes the environment, the air that we breathe, infected individual that we are around or come in contact with, contaminated food and drinks, also again, hospital personnel. Therapeutic regimen. Drugs used to treat infections may alter the body's way of defending itself. Again, recall back in my lecture, are antibiotics good? Well, yes, they are. Yes, they are. Yeah.
You can't drink with them, so it's bad.
But you can, yeah. But there is such a thing as abusing it. So if you are antibiotic dependent, when you get sick, you go to the hospital or the doctor, give me an antibiotic, and then you're well, and then you get sick again, you go to the doctor, ask for an antibiotic. Every time you go to the doctor and ask him to prescribe you an antibiotic, you may be producing superbugs in your body, because whatever's left, whatever survived, remember what they say, the strong survive. So you taking those antibiotics may actually be a bad thing. You're increasing your threshold. And then pretty soon there may not be an antibiotic out there that will cure your illness. And you're hoping that your immunity kicks in. And then the opposite is also true. If you are given some antibiotics, and it's, let's just say, for example, they give you a prescription to take, one pill a day for 5 days, but you start feeling better at the second day, you're going to stop taking it. Okay? And so again, that's another problem. The moment that you stop, whatever survived, you're giving those bugs an opportunity to get stronger. So those are the two extremes. Equipment, dirty equipment and instruments is also can cause nosocomial infections. Contamination during a medical procedure, a break in sterile technique in which infectious organisms are introduced. For example, through a wound, performing different types of invasive procedures. What is your number one protective barrier? It's going to be your skin, right? So your skin. So anytime there's a break in the skin, again, recall it's not just the skin, outside skin, but we also have inside skin, the protective membrane of your tissues and organs. So if there's a compromise in that, there's a chance or risk of getting an infection. So invasive procedures, breaking the skin, such as surgery or performing angiograms. The use of long-term appliances such as PICC lines and urinary catheters. Are you guys used to these terms now? You've heard of PICC lines at the hospital? So basically it's just an over-glorified IV that sticks out of your body for days, weeks, months, or even up to a year. Regular IVs should be only in you for a day maximum, sometimes 2 days. But a PICC line can persist for a very long time. And so now the problem with these long-term appliances is that it is trapped inside your body. Anytime there's communication from the outside environment and the inside environment, you're increasing your chances of getting an infection. So those who have PICC lines, what do you think they are taking to prevent an infection from having that catheter in them? Antibiotics. It's catch-22, right? All right, so social and economic factors that contribute to the risk of getting some type of infection. First one here is the age. So if you're young, if you're old, there's a greater chance of you getting an infection because when you're young, you're building your immunity. When you're older, your immunity starts to break down. Congenital and genetic diseases. So sometimes we're just born with poor health. Nothing we can do about that. Nutrition. Eating healthy. Things that have a lot of vitamins, lots of minerals. Those that will combat illness. Exercise. Exercise is important to minimize the amount of infection. Stress. Work, personal, financial, relationship. I should include their
Tests and quizzes.
Yeah. Yeah, let's put up their tests. Yeah, so stress can wear down your immunity. One of the biggest factors. Not having enough rest. Are you guys averaging about 6 hours of sleep a night? 7? 8?
6 is probably
6?
5 or
Okay.
6.
Okay. I average 5 hours of sleep a night. That's what I do. I do 5 hours. It used to be easy to do, but as I get older, it's getting harder and harder to do it. Personal habits, drug, alcohol, dangerous sexual practices, health history, including different diseases like diabetes, heart disease, chronic lung problems, and children who haven't been immunized. And then we'll talk about immunization later on. There's a big controversy that comes along with that. Is it good? Is it not good? There is data and statistics out there that proves both sides, so it's going to be in debate for a very long time. Inadequate body defenses, where there's a break in the skin from burns and or trauma, surgical and medical intervention, and therapeutic regimen. Again, anytime there's a break in the skin, you're increasing your chances of an infection. The bloodstream and the urinary tract are the most common sites for nosocomial infections. These often result from long-term use of vascular access devices like PICC lines, dialysis catheters, and portacatheters. Also retention urinary catheters all contribute to that. So again, our skin protective membrane is the first line of defense. You guys have seen this now, right? You guys are in the hospital, you've seen different kinds of catheters.
Yeah.
Okay, sticking out of the arms, the neck, the chest. So anytime there's a communication, again, from the outside and the inside, those microorganisms will track their way in and cause an infection. But again, it's necessary. The best of the two evils. By the way, do you guys know where this is done? Where a lot of this stuff is done?
Cardiac cath lab.
Yeah, it's done in radiology. Yeah, patients come out, when you guys get a chance to go into cath lab, but also special procedures, they're putting one of these things in. We do that. We do it under x-ray because we got to make sure that the end of these IV lines are going in the heart and not down the chest or not up the nose. Okay, so we need x-rays to do that. And you may be scrubbed in with the doctor to put those appliances in. Microorganisms that cause infectious disease is called a pathogen. They are characterized by their invasiveness, which is their ability to infect, their pathogenicity, how dangerous or lethal that pathogen is, their virulence in their ability to grow or multiply, and then their specificity, their attraction and susceptibility to the host. So those are the 4 characteristics of these pathogens. Now, we also know that microorganisms exist all around us. We have bad microorganisms, but we also have good microorganisms. But this one is important right here, is that relocation. So you can have microorganisms in their environment, which is naturally occurring. But the moment that they leave their environment, they can become hostile. And the example that I gave before is that of women, when you go to the bathroom, how are you taught to wipe?
Front to back.
Front to back. I had to think about the analogy. Front to back, right? So why not back to front?
Poopy.
Right, exactly, right? Because those bacteria, they normally exist inside your GEI tract. That's normal. But outside of the environment, now it becomes hostile. And so that's how you get UTI. All right. So again, normal, outside of their environment, can cause problems. You have normal, non-harmful organisms in treatment and sewage waste. They're used for brewing, winemaking, pickling, yogurt and cheese.
Yay.
Yeah, fermentation to produce ethanol and other liquid fuel. And also there's that new thing, probiotics. So we're trying to increase the good bacteria in our gut. All right, different classifications, going to keep it very basic. Of our microorganisms or pathogens, we have bacteria, fungus, parasite, and virus. Am I going too fast? No. Okay. So bacteria is a single-celled and prokaryotic, meaning that it lacks its true nucleus in organelles. Genetic material consists of a single loop of double-stranded DNA. And this is why bacteria is so bad, is because they can replicate, once exposed to it, they can replicate every 20 minutes. So when you have a bacterial infection, it can rapidly spread. Some are highly resistant, so if they have a protective covering or coating around them, they're known as an endospore. So it has like its protective shield. These spores are highly resistant to chemical and physical agents and to the external environment. So you have your bacteria, protective coating, that's known as an endospore. They can be viable and go dormant for many years, then germinate in response to a specific environmental condition. I can't speak. Okay, so that's bacteria for now. Mushrooms. Oh, fungus. I was thinking of
Same
steak,
thing.
I was thinking of steak and mushrooms. Okay, fungus. They can either come in a large form, macroscopic, such as mushrooms and puffballs, or microscopic, such as yeast and molds. They are eukaryotic, having a nucleus and organelles, and they exist in 2 forms. Those 2 forms, first one is a single-celled, like the yeast, and then you have the filamentous type. Every time I think of filamentous, I think of a sperm cell, okay, with its tail. Okay, so filamentous hyphae. Also, if they have many branches, it's called filamentous hyphae. So it's kind of like starts off real small and then branches out. And at the end of those branches is where you find the spores, the reproductive spores. So these spores can be easily transmitted in our environment by a simple wisp of the air. So they can be in one, confined to one area, but all it takes is just a little bit of wind and it's now dispersed to other areas. Okay, any questions? Parasites, organisms that live on or in other organisms. It can be a plant or an animal. We'll just cover two basic types. One is the protozoa, the other one are helminths or worms. Protozoas are single-celled organisms. They are eukaryotic, having a nucleus and organelles. Compared to a bacteria, they're much bigger, much larger. And just like the bacteria, they may also have a protective coating around them. And when they do, they're called cysts. Now the thing about cysts is that they can thrive and live outside of the host. They don't have to be in the host to live, they can be outside of the host. So that protective coating is what's going to allow them to survive. They may have parasitic characteristics where they are able to ingest food and particles and have a simple digestive system. And then also here, they are described by their motility. If they have little projections, hair-like projections around them, that's the cilia. So I think of it of the eyelashes around the cell, like this, eyelashes. Or it can be also move around by flagella. Again, whipping tail-like action. I think we talked about protozoa last a couple weeks ago, right? When we were talking about Giardia. Yeah, briefly, Giardia. Okay, helminths, parasitic worms that usually live in human intestinal tracts. You have the flatworm, the platyhelminths, and then you have the roundworms, the achyhelminths. Some other type of helminths include the pinworm, tapeworm, and also trichinosis. But as technologists working in a hospital, we're concerned with bacterial infections, and this is the other one, viral infections. Okay, those two are very aggressive. So the virus is a simple organism, neither prokaryotic or eukaryotic. They are obligate intracellular parasitic-like viruses, organisms that cannot live outside of the living host cell. So viruses carry their own genetic information in either DNA or RNA. It's one or the other, never both. Like humans, we have both. For them, it's one or the other. So what these viruses do is they attach themselves to a host cell. They inject their code, their information on that cell, and that cell has to follow whatever that code says. It takes over. Kind of like a computer virus. That's where the term came from. Okay, because a virus goes in the computer, the computer doesn't do what you want it to do anymore. It's following somebody else's instructions. So that's what a virus does. And now after the virus has used the cell dies. Okay, and then they transmit their information to other cells. Okay, so any questions about the virus? A lot of this stuff reminds me of my ex. Some of you guys agree, right?
Yeah,
We've been there. We've all been there, right? Yeah. Most of us have been there.
I think it's a parasite.
The parasite?
Mommy. Forgive me.
Okay, elements of transmission, the cycle of infection. So we'll cover the steps in the cycle. So we'll talk about the host, the reservoir, an exit portal, how it's transmitted, and then an entry portal. So the first one here is the infectious agent. The infectious agent is either using One of these hosts, it can be the sick or disease, they can be living or dead. And then you also have the carrier. A carrier, as we'll talk about that later on, but a carrier is someone who has an infectious disease but isn't presenting with the symptoms. So here are some examples of carriers. So again, healthy individuals having these pathogens living in them, but they have no idea because they are asymptomatic. They're not demonstrating with the sickness. So this includes Staphylococcus aureus. It's generally a skin type of infection. It's bacterial. It can cause pneumonia, cause food poisoning, toxic shock syndrome. This is right here. This causes septicemia, and death can happen within hours to a couple of days. Same thing with blood poisoning or bacteremia. And then we have Typhoid Mary, who was a healthy food handler. She was contaminating foods that she cooked and serving it to her, to the families in which she served. She used to be a housemaid, a house cook. Now, have you guys ever heard that story? Mm-hmm. Okay. Yeah, so back in the 1920s, I believe it was, she was a cook for a prominent family in upstate New York. And so she had typhoid. And the way typhoid is transmitted is just, you're touching your face, you're touching your body, and then handling the food. So that's how it was being transmitted. So again, she was asymptomatic. She didn't know she was sick. So she would serve this food to the family. They got ill, and they thought it was isolated. And then there was a huge epidemic of people showing up to the hospitals with the same type of symptoms. And they're thinking, well, where is the relation here? So back in the day, they didn't have the CDC, they didn't have OSHA, they didn't have JACO. What they had were detectives, or real police officers trying to find out how this happened. So what they did find out was that she wasn't just a cook for one family, she was a cook for many families. A maid for many families all of upstate New York. So that was the link. So when they found out it was her, they told her to stop doing it. And she did for a while.
She tried to put him on her own little island by herself.
Yeah, so yeah, they tried to isolate her. Well, that was the second time, I think.
Yeah,
Because during the first time she was arrested, they did imprison her, they told her to stop, and she said she will. But hard times, right?
She was like, oh, I'm not sick.
Yeah.
You know,
So she
couldn't
was in denial.
prove that she wasn't sick.
Right. She was in denial, but also she needed to make some money. So she continued to do her work, and then eventually they, I think, arrested her for good. Yeah, imprisoned her for good.
They tried to put her on the island, and then she tried to escape. Her brother came and got her or something like that. Yeah, they actually put her on the island to live there on her own, and she left.
Well, you know more about her than I do. Yeah. Okay. So did she ever escape?
Yeah, she got off the island, and then she started working for other families, and they found her again.
Oh. So she didn't stop. Okay. All right. So typhoid, bacterial, causes fever, skin rash, lethargy, extreme abdominal pain, diarrhea, vomiting, and then respiratory distress and brain swelling. I think there were maybe only 2 people that died from it. A lot of people were affected by it, but I think only 1 or 2 died. Oh, you don't know.
I just
Okay, you know.
they're digging her on the island
Okay.
there, so.
All right. And then HIV, human immunodeficiency, is another type of carrier disease. So you guys all know Magic Johnson, right? Magic Johnson, former Laker player, Hall of Famer, entrepreneur, was diagnosed with HIV, what was it, like 30 years ago? A long time ago. He's still alive. Yeah.
Infecting
alive.
others.
Huh?
Infecting others.
I don't know. I don't know. So, and you guys know my own opinions about that, right? You're telling me there's no cure for this? You're telling me there's no cure for cancer?
If you have enough money. Yeah.
There's cures out there, yeah. But the thing is, again, if the cure was out there, guess who's going to go broke?
Medical
Pharmaceutical companies.
institutions.
There's no more business, and the economy goes to crap. Are you recording me?
Yeah.
Don't repeat that.
It was a
Those are my
joke,
opinions.
joke.
I'm sure there are cures out there. They're just not being divulged. That's just my opinion. With the technology that we have, come on.
Uh-huh,
Nanotechnology, come on.
exactly.
We got the technology. We can't tell me there isn't a cure.
Yeah.
Okay. Number 2 is the reservoir of infection. So the source of the reservoir of infection is any suitable place where pathogens can thrive in sufficient numbers to pose a threat. So microorganisms like to live in warm, dark, moist places, and as long as there's plenty of nutrients, they can thrive. So warm, dark, and moist, and plenty of nutrients. The source of infection can be a patient with hepatitis. It can be you and I with an upper respiratory infection, or a linen handler. Remember, staph, skin. They're rubbing up against the linen and then laying the linen on the bed. So linen handler with Staphylococcus aureus. Humans are the most common reservoirs of infection. Secondary sources includes contaminated foods, water, or any damp, warm place that is not cleaned regularly. The exit portal from the reservoir. So the pathogen needs a way to leave the host. So that can be by way of nose, through the mouth, the intestine, the urinary tract. If you have a paper cut, an open wound, remember your skin is a barrier. Now those pathogens can escape that barrier and infect the next person that they come in contact with. So these are just examples of portals. The fourth one here is the means of transmission. So when we're talking about the cycle of infection, this is where we intervene. This is the best way to stop that cycle of infection, is by stopping their means of transportation. So there are basically 4 basic types of transmission. We have 5 here because droplet and airborne kind of fall in the same category. So you have direct contact, indirect contact, droplet, airborne, and also vector. Have you guys gone to do a portable examination? Okay. And then right outside of the door, there's a sign that says isolation, and it tells you what type of isolation it is before you enter. So it'll tell you what type of isolation it is, and they'll also tell you what to wear to protect yourself. Okay? So the first one here is direct contact. This is spread when disease-causing microorganisms pass from the infected person to a healthy person by direct physical contact with blood or bodily fluids. Direct contact. Examples of direct contact includes touching, kissing, sexual contact, contact with oral secretions, contact with body lesions, and also a needle stick. So this is person to person, direct, person to person, direct contact. Got it?
Mm-hmm.
The next one here is indirect. Indirect. So spread when an infected person sneezes or coughs, sending infectious droplets into, this is just an example, sneezes or coughs, sending infectious droplets into the air, landing on some kind of inanimate object, and now the healthy person comes in contact with that contaminated object, and they start rubbing their face or licking their fingers or whatever, okay? This is indirect. So it comes from an infected person to an object to a healthy person. So it's person, object, person, indirect. Person, object, person. This contaminated object is known as a fomite. The contaminated object is known as a fomite. So touching objects that have been contaminated includes an X-ray table. Again, if you did not properly disinfect it, or not disinfect it at all. The X-ray table is a fomite. Wound dressings, clothing, medical equipment and also instruments are examples of fomites. Okay, but the key here, indirect, is person to object to person. Okay, there's an object that's involved. Well, what happened back here? Here we said it was person to person, but what is this? What are those? What is this? It's an object. So this contaminated object, what is this called? It's still called a fomite. But here's the exception. Dirty needles, contaminated needles. So infection by a contaminated needle is considered direct contact, a direct infection. Because the way they see it, whoever came up with this ideal of calling it direct is coming from one person and into another person. Okay, the needle is going from one person and then going into another person. That's why they call it direct. That's the concept. So again, this is the exception of an object. Okay, is the needle still called a fomite? Yes, it's still called a fomite. All right, so that's indirect. Next one here is droplet contact. Refers to infectious secretions coming from the conjunctiva, the nose, or the mouth. Have you guys ever seen these slow motion videos before of someone coughing or sneezing? So examples of droplet contamination often occurs when an infectious individual coughs, sneezes, or even speaks within your vicinity. And this is why, you know, like when we're speaking, I try to keep a distance away from you guys. When I'm lecturing, I'm not up in your face because I spit when I talk. And same thing, you don't want me spitting in your face, right? By the same token, I don't want you guys talking and spitting and your spit landing on my lips, and I'm like, you know, I don't want to do that. So what they say is for safe distances, when it is droplet contact, is a minimum of 3 feet separation. Minimum of 3 feet. Okay, that's what droplet.
Meaning you need to keep 3 feet separation, right?
Yeah, you and I have to be at least 3 feet away from each other. Anything closer than that, then that's direct contact.
For extra credit.
Any questions for Kat?
No, thank you.
All right, airborne transmission. So I said again, kind of droplet and airborne kind of fall in the same category. So airborne is referring to droplets that have been evaporated. Okay, so if they're evaporated, they become airborne and can travel greater distances. They can travel greater distances. Okay. And then vector. Vector is any living carrier that transmits an infectious agent. It's usually insects. Could they be plants? Yes, they could be plants. Okay, but they're usually insects. Mosquitoes, for example, that transmits malaria, fleas that harbor the bubonic plague,